This Embryology Highlight screencast will review mitosis, meiosis, and gametogenesis. Before discussing mitosis, let's review some features of chromosomes. On the left, we see that chromosomes are composed of double strands of DNA. Chromosomes are uncoiled when they are being transcribed and when they are being duplicated prior to cell division. In preparation for mitosis, these elongate chromosomes become greatly coiled. They wrap around histone proteins, and they undergo multiple degrees of coiling to form these visible chromosomes. So at the left here, we see this process of coiling. Down here, we see a drawing of elongate chromosome wrapped around histone molecules, and these in turn wrap to form two chromatids of one chromosome. And here on the right is a scanning EM image of one chromosome. It has been duplicated during synthesis, so we have two chromatids. These two chromatids are joined together at a centromere, which is shown here. Prior to synthesis, this chromosome consisted of just one chromatid. Following synthesis during interphase, a duplicate chromatid was produced. This screen presents more information about chromosomes. At the top here, we see at the left a very high magnification of a double strand of DNA. At a lower magnification, we see this strand wrapped around histone proteins. Here we see, at a lower magnification, coiling of this arrangement forming a thicker strand. Following synthesis, we would have two duplicate strands which are joined by a centromere. Then prior to mitosis, these coil even more to form a typical chromosome. At the upper right, we see a single chromosome composed of just one chromatid, as might be found in a new daughter cell. And then during the interphase synthesis, prior to mitosis, this then have two chromatids making up that single chromosome. Following mitosis, we would have two daughter cells, each one having one of these chromatids, and this process then would be repeated through multiple mitoses. Below we see karyotypes of human chromosomes. At the left is a photograph, at the right is a drawing. Each chromosome here consists of two chromatids because these karyotypes were obtained on a cell that was about to undergo mitosis. This is a diploid cell, and so the chromosomes appear in pairs. One chromosome was maternal, the other chromosome was paternal. The maternal and paternal chromosomes are matched. We have matched pairs except for the X and Y chromosome. So this karyotype is from a male cell. This diagram suggests a life cycle for a individual. This individual begins life as a zygote, a single cell. It proceeds to be billions of cells through mitosis. The initial mitoses occur in the embryo, and subsequent to birth, the adult grows by means of mitosis, and mitosis then produces germ cells. Germ cells undergo mitosis. They produce primary spermatocytes and oocytes that reproduce gametes by meiosis. So mitosis produces genetically identical cells. Meiosis produces genetically unique gametes. This diagram illustrates a cell cycle, and it shows that following mitosis, which produces two genetically identical cells, the cell must undergo interphase before it can reproduce again and cycle back and divide via another mitosis. And during that interphase, then, there is a synthesis of DNA. So at the start of that interphase, there's one chromatid constituting a chromosome, And following synthesis, two chromatids constitute a chromosome. This diploid somatic cell then can divide and go through the cycle again where each daughter cell has one of those chromatids. This mitosis cycle is typical for somatic cells, but also germ cells, oogonia and spermatogonia, also reproduce by mitosis. This diagram will illustrate mitosis. We begin with newly formed daughter cell, which has total of four chromosomes, two from paternal and two maternal. This cell then undergoes a process of interphase during which there is a synthesis, and following that we have two chromatids per chromosome. So we still have the four chromosomes, but we have two chromatids per chromosome. If we look at the process of mitosis begins with prophase, during which the chromosomes now become visible. They're not visible until they coil up to begin mitosis. During metaphase, the chromosomes align at the equator individually. They're not paired up. They're all individually aligned at the equator. During the anaphase stage, one chromatid of each chromosome then will go to each newly formed daughter cell, and you can see the beginning of a division of the cytoplasm. And so we end up with two identical daughter cells. Each has gotten one identical chromatid per chromosome. Finally, in telophase, we have a complete separation of the two cells and a reformation of the nuclear membrane. The chromosomes will eventually uncoil and then not be visible as individual chromosomes. So the point is that mitosis forms two identical daughter cells that are diploid. They have the same genes as the initial parent cell. They then will undergo synthesis and have two chromatids per chromosome. This diagram shows the cycle of a germ cell, which is capable of producing genetically unique gametes. It does so through meiosis, which is a two-stage process. It begins with a primary oocyte or spermatocyte that is a diploid cell, and then following meiosis 1, we end up with secondary oocytes or spermatocytes, which are haploid germ cells. Following meiosis 2, we end up with each secondary oocyte or spermatocyte producing two spermatozoan or oocytes or ova, each one having just one chromatid per chromosome. So we start with one cell, we end up with two secondary oocytes, and each produces two gametes. This diagram will highlight meiosis 1 division. It begins with a germ cell, a primary spermatocyte or oocyte that was produced by mitosis from a spermatogonium or a oogonium germ cell. In this particular cell, we have four chromosomes, two paternal and two maternal ones. Following a synthesis phase, then, we begin prophase of meiosis 1 with still four chromosomes, but each now has been synthesized, so we have two chromatids per chromosome. And the thing that's unique about meiosis is the fact that these chromosomes pair up. Here we see that the linkage during metaphase, what happens is they line up at the equator of the cell in pairs. So instead of lining up individually like they do in mitosis, they line up in pairs during meiosis. And because they are paired, it's possible and common for there to be crossover in the sense that maternal and paternal chromosomes will exchange DNA. And this creates genetic diversity. This pairing during meiosis creates genetic diversity. So whereas we started with identical chromatids per chromosome, we have, following crossing over, chromatids that are not identical to one another. The process of meiosis 1 proceeds with one chromosome of a pair going to one daughter cell and the other chromosome going to the other daughter cells. And this process is sort of random, so a particular daughter cell will get some paternal and some maternal chromosomes, and the other daughter cell will get the other paternal and maternal chromosomes. So the two daughter cells are genetically unique, and then during telophase we end up with two secondary haploid spermatocytes or oocytes. This diagram shows crossover during meiosis 1. We're talking about one chromosome pair, and one of the chromosomes of the pair is maternal, and that's shown lighter, and the other is paternal, which is shown darker. And following synthesis during interphase, we have two chromatids per chromosome. Each chromatid is identical, but during meiosis, these chromosomes will pair up, and so we have the opportunity then for them to exchange genetic information, which they do. So the paternal and maternal chromatid can exchange genetic information. And here we see a case where chromatids have exchanged genetic information. And so where we had genetically identical chromatids per chromosome, here we no longer have that. So this chromatid is not the same as this one anymore, nor is this chromatid the same as that one anymore. And this illustrates how they're different. So we start up with the paternal and maternal chromosomes. During the pairing up, they exchange DNA, where we started off with two chromosomes with identical chromatids. Now we have two chromosomes, and each chromatid is genetically distinct. So this diagram summarizes meiosis 1 and meiosis 2. We begin following synthesis with a primary oocyte or spermatocyte, where the two chromosomes, maternal and paternal, paired up. Then during that pairing they could exchange information, so each chromatid would not be identical as it started. That is not illustrated here, but we just discussed that possibility. So then during meiosis 1 we end up with two daughter cells. Each is haploid. They are genetically unique because each daughter cell has gotten a different combination of maternal or paternal chromosomes. Then, as we proceed through meiosis 2, we see that meiosis 2 is similar to mitosis in the sense that the chromosomes align at the equator, and one chromatid will go to one daughter cell and the other chromatid of a pair goes to the other daughter cell. If this were mitosis, these daughter cells would be genetically identical, but because the chromatids are not identical, even though that's not shown on this image, each daughter cell then gets a chromatid that's not identical because of crossover, and so the two gamete daughter cells then, sperm or ova, are genetically unique. We have produced 4 genetically unique gametes from the one primary oocyte or spermatocyte. During gametogenesis, diploid germ cells give rise to haploid gametes through meiosis. In the case of males, the primordial embryonic germ cells are influenced by testicular Sertoli cells to become spermatogonia. In the case of females, the embryonic germ cells do not have the Sertoli cell influence from the testis, and so they then by default become oogonia. Let's consider first the male situation. Following puberty, spermatogonia give rise via mitosis to primary spermatocytes, and this is a continuing process ongoing in the sexually mature male. The primary spermatocytes then, which are diploid cells, undergo meiosis 1 and produce haploid secondary spermatocytes. Those secondary spermatocytes give rise via meiosis 2 to spermatotids, and so one primary spermatocyte gives rise then to 2 secondary spermatocytes, and they give rise to 2 spermatids, so a total of 4 spermatids are produced by 1 primary spermatocyte. Following the formation of spermatids, there is a morphological transformation to spermatozoa, and so they have to change their shape and develop tails. This cartoon illustrates the spermatogenesis that is ongoing in the mature male. Spermatogonia via mitosis produce primary spermatocytes. Each primary spermatocyte produces 2 secondary spermatocytes which are haploid, and each secondary spermatocyte then produces a spermatid, which then has to undergo a transformation to produce free swimming spermatozoa. What's interesting about this process is that the cell divisions are incomplete, so there are cytoplasmic bridges between these cells. And this shows a section through a portion of a seminiferous tubule. Here's the lumen where free swimming sperm will end up. Here's the outer wall. At the outer edge we have spermatogonia. They are reproducing, producing primary spermatocytes. They reproduce and produce the haploid secondary spermatocytes and produce spermatids, some of which are evident on the interior. In the case of females, oogonia via mitosis produce primary oocytes that initiate prophase of meiosis 1. But this process all takes place prior to birth in the embryo. Following birth, there is only primary oocytes in prophase of meiosis 1. They are surrounded by follicle cells, and these follicle cells suppress the continuation of meiosis 1. So these primary oocytes are in prophase of meiosis 1, suspended in that state. And then following puberty, there are selected follicles at each estrus period that are going to develop and lead to ovulation. It is at ovulation then that meiosis 1 is completed. Ovulation releases the oocyte from the follicle and it's able to complete meiosis 1. Meiosis 1 leads to a secondary haploid oocyte, and if that is fertilized, it will complete meiosis 1. So we end up with a zygote, a fertilized secondary oocyte. This shows a drawing of a bovine ovary. At birth, the ovary contains these follicles, these primordial follicles, which were produced in the embryo. Each follicle contains follicular supporting cells surrounding an ovum, which is in prophase of meiosis 1. And at each estrus cycle following puberty, a few selected follicles will become gravid follicles. Following ovulation, meiosis 1 will be completed. And if fertilization takes place, then meiosis 2 will proceed. This shows a oocyte that has been ovulated. What gets ovulated is the oocyte, its surrounding zona pellucida, which it has secreted, and then some surrounding granulosa cells. Each of these granulosa cells is a normal sized cell, so you can see how large the ovum is relative to these other cells. This is the nucleus of the ovum. It has some small amounts of yolk in the mammal, and so a sperm would bind to the zona pellucida and then release enzymes and work its way through to ultimately bind with the oocyte membrane in fertilization, and that would cause the oocyte, which would be a secondary oocyte following ovulation. That secondary oocyte would complete meiosis 2 with successful fertilization, become a fertilized zygote. Now, each primary oocyte that's in those initial follicles is going to produce 4 gametes. In each division, the cytoplasm will go with just one of the nuclei. And so all of the cytoplasm ends up with one of the gametes, and the other gametes consists of just a nucleus, which is discarded. It's called a polar body because it just sits off to the side, not evident in this image here. That concludes this embryology highlight screencast that reviewed mitosis, meiosis, and gametogenesis.